First speaker is Xavier Draye. He is from the University of Louvain in Belgium, and his title is Root System Architecture Beyond Geometry, New Opportunities or Undue Complexity.
Good afternoon, everyone. Okay, better. Thank you. Good afternoon, everyone. It's a real pleasure and to be here, and I would like to thank first the organizer for the invitation, and second, I would like to thank Andrea Carminati this morning, who made a wonderful presentation and introduction to what I'm going to talk about, and also for the following speakers. Okay, I have to learn. Right. So when it comes to the efficiency of capturing water, I would like to start with the basics and quickly go to the ultimate. So it's kind of easy to figure out that that's the general picture. Water move from the soil through the roots, and then before it reaches the shoot where it is transpired. The complexity comes from the fact that water moving along its path is going to very different sort of materials and will be driven by different type of forces. Andrea Carminati explained very well this morning this part of my slide. So showing that on its way from the soil to the root surface, the water can fill or traverse medium where the radial conductivity, well, the hydraulic conductivity will be dropping in transient condition. So the fact is that the similar phenomenon can occur on the next path of the water. So when the water is actually crossing the root tissue, it will go through three parallel pathways. So going through the cell walls, through membranes, through plasmodesmata, and each flow will be determined by different gradient forces and also different conductivities. At the same time, those conductivities can be regulated in time by the plant and by the cell, so that going through this medium is again something which is quite complicated and will show transient variation over time. We also, it was mentioned this morning, have transient phenomenon that can occur when water is moving through the xylem vessels. And obviously, one of the points that I have to spend some time on it, because the talk is about root geometry in architecture, is the fact that all those processes, all those phenomena will occur in different ways along different routes. The root type will matter, the position into the soil will matter, and the connections between these elements will also make a difference. And this is where root architecture comes into the play. So we need an additional layer of an additional scale where all these phenomena will be aggregated. Now, one of the big questions is, do all of these phenomena, are they all equally important when it comes to talk about water uptake in drought-prone environments? And I'm going to, so Andrea and Steve will speak about this, and I'm going to talk about the architectural part. So one way to explore and to make the sorting is to get inspiration from what people developing crop model have been actually doing. They have made a selection of what is important and what is less. And what we find in the crop models, and this is a case of the R.SWMS crop model, is that root architecture is basically degraded to the vertical dimension, a little bit of the horizontal also, through a factor which is called an occupancy factor. It also captures some of the soil hydraulics through a term which is a conductivity of the different soil layers. But that means, and people working with these models seem to be happy with what the model predicts. That seems to indicate that it's only a matter of root depth and soil water release. Now, trying to go into more details, Jonathan Lynch and colleagues have been the ambassadors of a number of efforts trying to capture a little bit more of the complexity of root architecture in sort of an optimization exercise to try to figure out if we can imagine a root architecture phenotype that would maximize the investment, the uptake of water for investment of carbon. The principle that they use in this type of work is basically exploit the fact that the root system can be dissected in different root types, and every root type can have specific growth behaviors and developmental behaviors. So basically what they can do is simulate a large number of root system architecture. In this case, you see that they have been keeping the seminal root system of maize similar and then changing the angle of the nodal root. And systematically for each of those virtual root systems, they can try to dissect how much of the soil is actually explored by this. And from that, they infer the amount of water that can be transpired. So this exercise has been done systematically by different groups for water, but also for nitrogen and phosphorus. And people came out with this type of nice illustration, and it's summarized in 3 words: steep, cheap, and deep. But this is basically the outcome of an optimization exercise. I'm not going into details, but you see that different root types, the similar root, you better try to keep them horizontal and long laterals, while the crown and nodal roots, you would have them deeper rooted. But the objective is, again, similar or consistent with what we had on the previous slide, trying to get roots deep in the most efficient manner. So in its paper, Jonathan Lynch ended up with a few open questions. What about the interaction about all these fins? The fins is different traits that characterize a root system architecture. And I would like here to step on the next, to step forward and to talk about new fins and new models to try to analyze how the things are actually working. So briefly, it's hard to avoid speaking about the methods when you talk about root system architecture and the context with breeding. So shovelomics seems to be a powerful technique with a reasonable throughput to capture the dynamics of topsoil root dynamics. And if you use it properly, you can even estimate the evolution of the number of, so the production of nodal roots over time, and compare different genotypes, and you see that you find some traits with some reasonable level of irritability. However, the steep, cheap, and deep ideotype is a matter of deep. So when it comes to predicting or to estimating deep roots, then we have a strong trouble due to the fact that deep root system architecture is highly imprinted by soil structure. You find basically roots flowing, growing into the large pores where water is also flowing. You observe this in soil condition. You also observe this when you do rhizotron experiments. So the idea is that deep root system architecture, if we cannot estimate it directly, we probably could assess it indirectly via an understanding and characterization of growth constraints or plasticity and metabolic cost. So once the roots have reached the level below the chevelomics, we don't know what they are doing, but maybe we can try to predict what they are doing if we know how they feel, how they sense the environment, and how they react. So the first avenue, I've picked up 3 avenues in my, for my presentation. The first avenue is to explore in more details the dynamics of root growth. And to analyze this, we are using a phenotyping platform that will allow, with a high throughput manner, to have a very fine time scale resolution of the root growth of individual roots during periods up to 3 weeks. So the sort of results that we obtain with this setting is sorry. Okay, thank you. The observation that we made is summarized on this graph, where you see the evolution of root length over time. For individual roots, the 4 graphs are 4 different plants, and it is a little bit striking to observe that once the root is emerged, it will keep growing at a constant rate. So we have tested that by making, I haven't done anything. I have tested that by making a linear regression, and you see that the R square is generally close to 1, and this is the case for 10 of thousands of roots that we have analyzed with the system. So this is a little bit of a surprise, as we know that roots are highly responsive to their environment, but you have to remember this is aeroponics, so they are growing in non-constrained environment. So one of the conclusions is the elongation rate is established at the time of root emergence. Another one is that the roots are able to keep that growth rate despite the fact that the plant is establishing new roots and growing new leaves at the same time. So a possibility is that the system is geared to anticipate subsequent assimilate expenses, or maybe allocation rules, like all the roots keep priority so that the growth rate of the next root is adjusted to what is available at the moment. But clearly, this was a surprising result for us. And then on the opposite, if you look at what lateral roots are doing, you observe that lateral roots display a large intrinsic elongation rate variability. If you have been looking at roots carefully, you see usually that you will find short lateral roots between long lateral roots. And so Sixtine Passo and Beatrice Moreno in Montpellier have been analyzing that systematically, tracking roots over the lifetime. And what they observed, so in a population of lateral roots that was done in millet, that was done in maize, is you can classify, so that's the evolution of the elongation rate. You can classify those lateral roots in population of subgroups that grow and accelerate, grow and keep growing, ungrow, and stop growing quite easily. To all of these 3 groups corresponds to different diameter classes and also different anatomical features like the number of xylem poles. The second avenue I would like to explore is the developmental plasticity, that is the response to external cues. And I will explore this with a story that was initiated by Tristan Lavigne, then taken over by Aurélie Babet, who observed that was an accident when growing plants in aeroponics. If you stop spraying the roots for a short period of time, you observed a region of the primary root that is devoid of laterals. So they were able to block lateral root formation. So that was not the goal of the study, but we have there a very nice system to do transcriptonic analysis and analyze the development of lateral roots, which we did. But part of the process was to characterize where this lateral root repression occur. And what we figure out, these are the different stages of development of lateral roots, that this was occurring before the first asymmetric division. So that was a very early response to this stop of lateral roots, stop of brumization. So Beata Orman took over this, and starting from the transcriptomic work, she could pick up genes that indicate a role for ABA signaling in that. So she translated that system to Arabidopsis, and using the genetic tools available in Arabidopsis, she could pick up ABA signaling genes that are specifically involved to this response. So now come to the important question. We seem to have a system developed and used by plants to block lateral root formation in a matter of minutes. So it takes less than 1 hour to see the complete response established in an irreversible way. So we have been discussing with a number of people to what would make sense in a root to reply and to respond so rapidly and in an irreversible manner. So that's the hypothesis we have come up with, is basically when a root enters a macropore, it loses the contact with the soil, it would stop lateral root formation and reinitiate after. There are many mechanical reasons why you would stop doing a lateral when you enter a macropore. I'm not going into these details, but we had some information that sometimes you see these sort of things when you do soil trenching. As you see here, a root that is growing into the soil, entering a pore, then leaving the pore, and you see that there is a block of lateral root formation. So we have made a collaboration with Malcolm Bennett and University of Nottingham, who could reproduce exactly the same phenomenon by growing plants in a soil in which you have made a little core, and you see with the micro-CT technique that the root is really stopping lateral root formation and starting again. So I believe that much more is going to come from developmental plasticity. And there was a nice paper by José Dinoni recently that shows a number of phenomenon like root response, growth response, or formation of apoplastic barriers, or hydropatterning, where you see that the root hairs are produced, and the same for lateral roots, asymmetrically depending on the moisture environments faced by the roots. You also observe some the hydrotrophism phenomenon and the formation of parenchyma. Now the future will tell us to what extent these responses that are induced by moisture heterogeneity in the soil do actually make sense in terms of water capture at the whole plant level. But this is far from being evident for me. So the third avenue that I wanted to explore is the progress that we have made in the last years about the modeling of the soil root system. And really, the objective is to combine hydraulics to work on the specificals to include what occurs in the rhizosphere, as was exemplified this morning by Andrea Carminati, to use those models to simulate long-distance signaling, to address the question of fruit coupling. So we are really, we need to connect different things together. And so we are talking about multiscale strategies. I will use two examples, one on the root system architecture plus hydraulic, and the other one on the hydraulic of the cross section of the root. So the first example, and I will explain the model by showing basically a simulation that validates the output of the model, is, well, that's the example. You see, this is a rhizotron used in a system called light transmission imaging that gives us a map of soil water content in real time. And this is over a little bit close to 36 hours. So the root system didn't grow much during that time. It was already exploring the complete rhizotron at the beginning of the experiment. And it's just a dry out experiment. And you see that water disappear, mostly close to the roots, to the shoot base, and then gradually extends to larger regions. So what the model is doing is simulating, we have a complete representation of the root system architecture with some assumption of the hydraulics between the different roots and the radial hydraulics. And the model takes into account the connection with the soil and the non-linearity of soil hydraulics, as was explained this morning by Andrea Carminetti. So After some fitting, because the hydraulic parameters are not known, so we can play with the hydraulic parameters until we find a best prediction of the model in terms of soil water content. So after fitting, we find some reasonably good simulation of the evolution of the soil water content, which is sort of a good news. And then the model will also give us interesting variables. And one of those is the sink term, so where water is actually taken up by roots. And you see that from the very beginning, water was actually taken up everywhere very homogeneously. And this occurs over time, except at the end where part of the soil was actually dried out. So our intuitive interpretation that water was taken up close to the shoot base, according to the model, was not correct. Water was taken up everywhere, but it's close to the shoot base that we see the drop of water content most easily. Now, predictions from this soil root hydraulic architecture are very interesting if we compare the ratio between the radial conductance, which represent the easiness of water flow through the composite transport system, through aquaporins and membranes, and the ratio of that radial conductivity with the axial conductance in the xylem, which is determined by the number of diameter of xylem. And what we observe from simulation is that if you switch to a low Kr over Kx ratio, which means that you decrease aquaporin expression or you increase the number of size of the xylem, the sink term, so the uptake of water will be more evenly distributed over the depth in the soil profile. While if you increase aquaporin expression or if you decrease xylem size or number, you have a massive shift of the water uptake to the upper layer in the soil. So you have to think about the consequences of that. You remember that Andrea was speaking about the speed at which water is actually flowing from the soil to the root surface. On this aspect is a very sensitive and fragile system because you force water to quickly move into the soil, and so you may increase the likelihood of those water conductivity drop into the soil. Now, another prediction of the model comes in if you consider one of the outputs, which is the xylem water potential, which is one of the outputs of the model. That shows you that if you have low Kr over Kx, so low aquaporin expression, large xylem vessels, you basically observe a constant, nearly constant xylem water potential across depth in your root system. That will decrease over time during the dry down, while if you increase aquaporin expression or reduce the xylem vessel, you have essentially the top part of the root system which fills the drought into the xylem vessels, while if you go down, you see that the roots feel the same sort of water potential. So in looking at the system from that perspective, you have the feeling that this scenario would actually save, if you take this into consideration with data that show that the root growth rate of a root is affected by the local soil water, the local water potential. This suggests that this system would actually preserve root growth towards deeper layers because the deep roots are less affected or less sensitive to this signal. Now I'd like to go deeper into tissue scale properties and to have a look at and we are designing at the moment a model that should allow us to estimate and to predict the radial conductance, which we have seen is a critical parameter, based on all what we know regarding our current perspective, our current understanding of water flow across a root section. So this is a close-up that shows the roots, the model that represents every single cell wall element, and the color indicate that the flow is, in this case, in this simulation, faster in the radial walls than it is in the tangential walls. You also observe the color of the membranes that indicate how fast is the flow through the membranes, that is through aquaporins. And the little holes here shows you the intensity of and direction of flow through the plasmodesmata. So all these processes are taken into consideration. And the good news is, so we have been, that's the work of Valentin Coureur and Marc Faget. Valentin has been looking over the literature to find values of plasmodesmata density, plasmodesmata conductivity, cell wall thickness. And so all these data from different work come together in the model, and you see that that's the different values here are the prediction of the radial conductivity as a function of the distance to the tip. And these are the values that have been measured. So we are incidentally in the same range of what would come up from the prediction. So what will this model tell us? It will allow us to establish good hypotheses about understanding, for example, the contribution of aquaporins. So you see here the distribution of the flow of water from the root surface to the xylem. So it is massively through the apoplasts and then recruits this symplastic pathway only when it comes close to the endodermis. And when the endodermis is fully developed and you have also an exodermis that is developed, then you see that the aquaporins would be more massively recruited at 2 different times. So this is a sort of prediction that you can get from the model, and that will drive you to look into the data, into the experiment, to see whether this is how things that are happening. So now I talk about multiscale. We had models with 2 different scales here. So we are now using this, and I encourage you to look at the poster of 16 Passo in the poster session. So we can now couple the information of lateral root growth variability into a model of root system architecture. And at the same time, we take the data on xylem vessel number, cell walls, number of the radial anatomy in the tissue model, and we couple those 2 information into the R.SWMS model. That allows us then to simulate, and in this case, that's the water potential and the color of different scenarios. So the purple one, the cyan one is the reference scenario. If you increase the proportion of roots that grow, of laterals that increase that fast, so that grow fast, so the red one, you get this green one, so you didn't increase the system performance. But if you stop, if you decrease their number, then you have a system which is highly sensitive. So that relates a little bit how the root growth dynamics needs to be considered together with the hydraulics before you can get a sense of what it could be. So I would like to just 2 slides on the real-life tests on the root depth contribution. This is a work of Nathalie Voigt and Adrien Dobbs. It comes from the DROPS project, where we had an inclined field experiment in Bologna, where we could replicate the same genotypes at different position along the slope. So these different genotypes were exploring different water depth, which is a really cute system. And we used thermal imaging combined with leaf area measurement to have sort of a proxy of transpiration. And we find, so the system was generating very different scenarios of water supply, but we find this very nice correlation between grain yield or plant height with the proxy of transpiration. So based on that, it's a little bit, yeah, we have the biophysical constraint and what? The interesting thing comes when you start to exploit the fact that you have those genotypes at different depths. So behind this common response, you find completely different behavior. So you have a genotype like MS153 that doesn't matter, doesn't bother. So while you have other ones that have strange response, so they have a yield response, but they don't actually respond in terms of that's a proxy of stomatal conductance. while you have other ones that are more sensitive in terms of stomatal conductance, but have sometimes a slow response, a short, smaller response in terms of heat. So the goal of this is really to remember that root is nice, root is fine, but if you look at root without looking at shoot, you're probably missing most of the story. And I'm there at my concluding slides, still 45 seconds. So I've been hesitating a lot. I apologize for that. I was a bit stressed on the root system architecture and how to better capture deep root system by taking into account growth dynamics and plasticity. And there are several posters on this topic in lateral and the root function. Yeah, the take-home message is there is certainly much more than root length density as a function of depth. Talk about rhizosphere conductance, hydraulic regulation. I didn't spoke about multiple stresses. I didn't spoke about the genetic variability of those traits, which is a critical point, of course. And we hope that the modeling and the progress in modeling will help us to bring the different parts together to try to make sense out of this. And I thank you for your attention.
Thank you very much, Xavier. I think thanks once again for keeping the perfect timing. You didn't waste a single extra